Testing repeatability. Environmental impact assessments typically use biotic indices based on macrobenthos. Compared to morphological identification, DNA metabarcoding of bulk macrobenthos is potentially faster, cheaper, and no taxonomic experts are needed. But first we need a standardized protocol that allows for reproducible and robust DNA metabarcoding results. Therefore, we sampled macrobenthic communities with high, medium or low diversity in the Belgian part of the North Sea and aim to solve 3 research questions. First, we want to know how many DNA extractions and PCR products are needed to capture most of the diversity in a sample. So in each location, we sampled 3 biological replicates, of which 6 DNA replicates were taken. One of these DNA replicates was used to assess variation between 3 replicates for PCR amplification. To pick up at least 80% of the species diversity present in the 6 DNA replicates, 3 to 4 DNA replicates were needed. And also for PCR amplification, at least 3 PCR replicates were needed. Second, we tested if a fixed DNA metabarcoding protocol is repeatable across different institutes. Therefore, subsamples were taken and distributed to 4 different institutes across Europe. These were processed using the same standardized protocol. In each institute, we see high to low number of species for high to low diverse location. And this pattern is very similar between the 4 institutes. Also, beta diversity patterns were very comparable, as we clearly see clustering based on the macrobenthic communities, independent of the institute that conducted the work. Finally, we want to know the effect of small changes in this DNA metabarcoding protocol on the alpha diversity. After small changes in the lab protocol were made by the different institutes, similar number of species were detected as with the fixed protocol, and between 70 and 75% of the species were shared between both protocols. This study shows that DNA metabarcoding offers a highly repeatable assessment of both alpha and beta diversity patterns, which support the suitability of DNA metabarcoding for the monitoring of marine macrobenthos.